can find out about what they're doing, but also to understand better what it is for them to be here and for us to learn from that. So good. So Kijun, please.
Hi, everyone. Welcome back to the afternoon session. So in this afternoon, we have this hot seat session. So we have 4 hot and new scientists talking about what's hot, what's new happening in their own lab. And then so each one of them will probably give a 20 minutes presentation. And then during the process, I would like you to prepare really tough questions. And then after the presentation, we will grill, and as you can see, we literally have a hot seat. So let's grill them on the hot seat after the presentation, okay? So our first speaker is Christy Burrows from my lab, Mathematical Biocomplexity Lab. And you take the stage.
Can you guys hear me? Okay. Welcome, everybody. Thanks for being here. I'm excited to talk about what I do because I don't get to do that very often because most people don't know what we do over here, right? So I'm really looking forward to this and to looking forward to seeing what everybody else has to offer as well. So this is not my slide. Let me see if I can do this here. There we go. This is me. Okay, so I want to give an introduction to maybe demystify a little bit about what we do, maybe at the upstairs in the Mathematical Biocomplexity Laboratory. So I should start off by saying that laboratory is a bit misleading. This looks a little bit more like our laboratory. It's not a wet lab, obviously, but we really only use the chalkboard. But what we do is work on problems in molecular biology as they relate to mathematics, and we impose our framework in mathematics onto these types of problems. So I want to detail a little bit about each of the individuals in our group. We have 9 people in our group, and then I'll give you a little overview of what we do, and then hopefully I'll motivate our problems that we do enough that you feel that our work is important. Okay, so we do two main things here. So we do a lot of theoretical mathematics to mean that we try to describe these molecular systems mathematically from a theoretical framework, but we also implement this in the form of software and algorithms. So that's how we make ourselves how do you say, applied in some sense, right? So these are our 2 main researchers, Phoenix and Thomas. And then this is me up on the top. This is the only picture that I had of myself within like the last year. This is Zion, not that that matters. But I'm doing my master's degree here. I'm in the math department, but I am here working on a combinatorial problem. Combinatorial just means counting problem. And then Mia is our undergraduate. So our group consists of 3 research scientists, 3 postdocs, 2 graduate students, and an undergraduate. And we are primarily from mathematical backgrounds, maybe somewhat of a combinatorial discrete mathematics background or computer science, but this is where we come from. So I want to detail a little bit about the current dogma of what we're doing, and then I will describe how we are trying to refine the molecular biology problem, the data problem, and make it maybe a little bit more, how do you say, statistically relevant, okay? So but let me first state our research objectives. So we're dealing with RNA and the process of sequencing data, and our goal is to incorporate a more structural information into this sequence alignment package that we get, which is sequencing DNA and trying to infer a phenotypic relationship based on the genotypic expression that we find in our data. And so what we're trying to do is incorporate a more structural information to mean that RNA has certain structure that we think implies certain phenotype, and we want to be able to give a more rich understanding of the sequences as they pertain to phenotypic expression. So the other thing that we want to do with this is to also deepen our understanding of evolutionary trajectories of sequences. So we want to talk about how a certain sequence can evolve into another sequence in a deeper way, as opposed to just this sequence is closely aligned to this other sequence. We want to really build upon this framework to make it more relevant to the problem of trying to find phenotypic expression in the genome. Okay, so the current dogma here, I think we all know. I'm pretty sure today in your biology, you guys aren't in high school, but I'm pretty sure that most high school students learn this framework in biology, in their freshman level biology classes, and it starts with the DNA, and the DNA pulls itself apart because it's a double helix, and then it transcribes itself into RNA, and then it folds and translates itself into protein. And so this is the central dogma that I think we've all sort of gotten accustomed to, right? And what we're trying to say is that this is not the whole story, and that we can deepen our understanding of this process, this molecular process, by understanding a little bit more of the pieces inside of these 3 steps. And I want to motivate this by saying that, first of all, in this system, in this framework here, we're assuming that the RNA translate into protein on the ribosome, right? But in fact, in the human genome, 98% of our genome does not translate into protein. And so we're missing 98% of the information. And we want to be able to build ourselves a different framework that incorporates this other really huge piece of information. And so up until, I don't know, several decades ago, it was thought that this 98%, and in other species it's a different number, but in this 98% of your genome was just junk. They literally called it junk RNA. And in fact, we discovered that that's totally not the case, and that there's so much cellular processes really rely on this other 98%, even though it doesn't code to protein. And so we can see this in gene regulation. We know that RNA that do not code to protein have a responsibility within the cell to regulate gene expression. And there also, we know about translational RNA that help facilitate the process of translating some RNA into protein. But there's a lot of stuff going on that's not just these 3 central points. And so what we want to do is expose these other areas that we think is like an untapped market of molecular biology. And we want to do that by building software that allows us to get into this kind of stuff. So this is maybe what something would look like. This is a piece of the genome, and this is folding. And so what I want to convince you of is that sequence alignment is not the whole story, that the nucleotides are not the most important part of the genome, but in fact how those RNA fold in order to make a cellular, something that the cell can work with, is even more important. So what I have here is a little strand of RNA that has been folded. And the point that I'm trying to make here is that in this first line, these 5 nucleotides on the left are paired with the nucleotides on the right. So we all know that RNA is single-stranded. And unlike DNA that's double-stranded, all of these nucleotides we call unpaired, right? And so what happens is that energetically, this RNA wants to fold in on itself, and it wants to pair with these nucleotides. And so what happens is that we then get this sort of tertiary or secondary structure out of this that is then used to inform phenotypic expression. And so what I'm trying to get at here is that there are a lot of different nucleotides that fold to the same thing. And here they're just flipped, but you could also have something different. And so there's not a one-to-one correspondence between structure and sequence. So we're missing a lot of this information, and this is what we want to work with. So the way that sequence alignment is going to date is the following. You take 2 sequences and you drop them in something. As you can tell, I don't know what this something is because I'm a mathematician, but you get these bits of sequence and you try to align them by inserting gaps and seeing, you just pair up the nucleotides and you try to see where these things are aligned. And this alignment then identifies this, so to speak, most likely evolutionary trajectory based on this cost of an evolutionary event happening. How close is this one to this one? How long does it take for this one to get to this other sequence, right? And so what we want to note here is that the cost of an evolutionary event is, so to speak, site independent. We don't necessarily say that at this nucleotide, this is the cost of getting to this other particular sequence. And that is the flow as of right now, right? This is sequence alignment as we know it today. And so what I want to convince you of is that by incorporating RNA structure into this framework and into this measure of sequence alignment, you can build a more robust model for going from genotype to phenotype, okay? So I want to give some examples here. Some of them are funny, some of them are maybe not so funny, but I want to give you some examples of why this framework is not necessarily complete at the moment. And the first example that I want to give is this guy right here. Oh, I just realized they have a laser. That's really fun. So here we have a sequence of genome, and we have this whole sequence is aligned except for at one point where there's the mutation that I've identified. And the difference between this one-point mutation is the difference between an entirely different phenotypic expression. So the top one is a healthy person, and I can't pronounce that other thing, but this other person has a different phenotypic expression. And so what we can say is that necessarily not always does a similarly aligned genotype translate to a similar phenotype, okay? Here's another example that we like to give that is particularly funny, that these two sequences, or these two sentences, if we can translate this into a grammatical problem, are extremely aligned, right? Their only difference is a comma. But if you were to take these two sentences literally, they would mean quite different things. Okay, so I'm hitting home the point here that this one-to-one correspondence is not necessarily perfect, okay? And here's another example. The converse is also not true in that a bad alignment does not translate to a different phenotypic expression. Because what I have here is two sentences. His success was due to his hard work, and his hard work made him successful. We would interpret these things grammatically as very similar. We could interpret them as the same thing. But if you were to just look at their alignment, they're not very well aligned. But in fact, we could metaphorically take this to mean that dissimilar genotypes can still produce the same phenotype. So hopefully I've convinced you that maybe something needs to change, at least somewhat. And so what we want to do is, instead of just thinking about sequence alignment and the nucleotides explicitly, we want to incorporate also the structure of these RNA. How do these RNA fold, okay? But what I want to mention, and what is a problem that's introduced into the evolutionary framework, is that one genotype does not fold to the same thing every time, and one genotype can translate into multiple, can fold to different structures, okay? And so this happens because, in general, energy, you have these energy models where you're trying to minimize the energy of this system as you fold, and this is not necessarily a deterministic process. But the same is true the opposite direction, which is what I've been saying, in that multiple sequences can fold to the same structure. And so what you get out of this is maybe a more robust framework for phenotypic expression, to mean that you can have these mutations and still preserve your phenotypic expression because your genome can fold to the same thing. And so what we want to do, and what the goal of our work is upstairs, I guess, is to consider these sequences and structures as pairs, and to find, instead of an alignment measure, a similarity measure that incorporates not only the sequence, but its structure, like I said. And we can quantify this, and we think that this could be extremely useful for people. And what I think was touched on earlier today in the presentations, and what I think is motivating our work, is that currently structures are not so much a part of the massive data sets that we're working with. And in fact, it's quite expensive to do crystallography, and it's time consuming, and I can see you're all shaking your heads like, yes, I know. But what we find is that when you have a theory, the technology will catch up eventually. And so if we can convince people that this framework is important, and then this structure is really important to our understanding of this molecular biology, then the technology will catch up, and we'll be able to do this sort of analysis more cheaply and more efficiently in the future, if what I'm doing here today is like convincing some people, right? So that's the biggest thing that we do. Oh, thank you. The biggest thing that we do is we try to design software and implement algorithms that will help us achieve this in the future. And the other thing that we do is consider the evolutionary trajectories of these sequences like in conjunction, in pairs with their structures. And what we can do is we define this neutral evolution to be evolving sequentially without changing your structure. That's what we call neutral evolution. And what this gives us is a correlation between the mutation rate and the binding information. But more importantly, what this gives us is a more robust way to do sequence design, because now you have a little bit of leeway in your sequences. So I think I can finish up here. The other thing that I want to mention is that these mutations and this cost of evolutionary trajectories can now be site-specific, to mean that the cost of changing a particular nucleotide that would inevitably change its structure is now incorporated into our understanding of this evolutionary trajectory, right? So we can take this new framework, and we can really build upon this in understanding the evolutionary tree of all this data that we have. Yeah. So let's wrap this up. I think hopefully I've convinced you that what we do is pretty cool. I think to summarize, I've said this so many times already, to summarize, we want to build upon this current dogma of molecular biology, and we want to incorporate this structural information. But we also really feel that this will lead us to more novel technology and more novel instrumentation in this way. Yeah. So I think we'll wrap it up. Thank you. Please don't feel like you have to grill me. I'm already very, very hot.
So since she finished her presentation, and it's about to grill her. So since I have a mic, I'm controlling the mic, so please allow me to have the privilege to ask the first question. And as we can see on I think the second slide, basically we are in the mathematical biocomplexity lab. We are a bunch of nerds that is getting super excited when we get an equation, or when we know how to count certain things. So I would like to ask you, and actually I would like to ask you this question for a while, is why do you choose to join a bunch of nerds like us?
So funny. Okay. I've always been interested in a lot of different things. And as an undergraduate, I think I had like 6 different majors or something like that. But I always found that mathematics was a critical component of all of these things. I can imagine that nobody else, maybe not everybody else, would feel this way, and that's totally fine. But I find that to have a strength and to apply it in ways in which another field maybe could help in this way, that I can more broadly apply what I know. And so in general, I joined this group because I think that it's a very unique group that is doing a lot of theoretical stuff, but in a very how do you say, like, directional. It has a lot of direction. A lot of mathematicians are just floating around.
That's a really straightforward answer. And any other questions? Please. Questions?
That would be a really great question for Phoenix, our computational guy. The answer is yes, Andrew. Yeah.
Your presentation was really interesting, and I have a question about when you say that you're trying to incorporate the structure in finding a similarity measure. What are the kind of measures or metrics that we can use in a structure? I mean, in the sequence, well, okay, you can understand, you can compute things like percent identity or whatever, a measure after you align them. But what can you use to, like...
a measure of if two sequences are similar.
Two foldings are similar. Or
two foldings are similar.
I mean,
yeah. Would we... Yeah, let me think about that. Kijun, do you have any...
Well, roughly, so there are there are various different similarity measure of structures. So you can do it in a very ad hoc way. So if you represent a secondary structure as arc diagrams, and then you can compare the difference of their arc diagram, like tree at the distance, or basically the based on any distance that's given from the level of graph, you can do it on the structural level. And also, if we consider the ensemble of structure of a sequence, then you can do a little more things. So, so basically, two sequences are the same if they are similar, if their ensemble are similar. And also, I think Reza is developing something using the Hausdorff distance that is mentioned in this morning's talk. That is also help us compute the sequence. And then, if you kind of like generalize the idea of considering sequence structure as a pair, then you can, in return, consider the ensemble of sequences with respect to a given structure. And then that will naturally give you a similarity measure on the structural level. And then ultimately, we just combine these two together. Any other questions?
Really, I'm the one facilitating this question and answer. I'm passing on the answers. The nucleotide content, you're saying? No, we think of these graphs as we think of nucleotides as vertices on a graph, and we lose that information. And really, what's important is the pairings. Yeah. Yes. Chris also has a question, Keisha. Chris also has a question.
Thank you. I think looking at the structure is a very interesting thing. As a biologist, I'm interested in the question like, how would you build a connection between looking at the structure and the phenotype? Because you said we not only look at the sequence information, and probably you can look at the structural information to say they might have the same phenotype. So how would you build that connection?
Yeah, I think the connection there is that the same structure would imply the same phenotype, right? And that a change in structure could maybe imply a change in phenotype. Is that your question?
I have two questions.
Okay.
I'll just ask both of them. So they might be related. So the first one is, so you come out of a math program, whatever it is, you're in a math program, and you're here. And so the first question is, in the course of doing this work, does this enrich the mathematical content of what you're learning? Is this just applying what you're already learning, or does this grow your actual mathematical training?
That's a really good question. Yeah.
The second one, and because it has to do with the value of these directed, this notion of the directedness has to do with the fact that this is an interdisciplinary institute.
Yes.
This is the experiential sort of aspect of a research lab. The second part of this question is we've heard about many times in your talk computational aspects of what you're doing. And so the question is, from a formal point of view, from a theoretical point of view, are you seriously engaged in looking at these things from a computational point of view, which is, after all, an area of mathematics, right?
Yeah.
So is that an area that you see how these two questions are
Yes,
related?
yes.
They're independent questions, but anyway, yeah.
I see what you're saying.
You got the questions?
I think I do. Okay. Yeah. So do you want me to talk about why this institution is more directed than, say, the math department?
No, I want you to tell me about whether
I remember your
mathematical your grounding is
Yes.
enriched.
Yes. Yeah. Okay.
And whether
yeah.
or not the computational theory is a part of this enrichment.
Yeah. Okay. To the latter question, not right now. My work is primarily mathematical away from the computational, but that's just because I have plenty to do in a 40-hour work week that I don't focus on the computational part of it, but that could certainly be a part of it, and my experience could also be enriched in that way. I would 100% say that my mathematical understanding is being challenged here, that I am learning new math here, even though my problem is biological, in the sense that I think there are things that you just can't learn in a classroom when you take an algebra class or something like that, and things don't fit together so easily. They're, like I said, they're just kind of like independent concepts that you're trying to just get put into a succinct textbook and try to put on somebody else. And maybe it seems, to me, it would seem really ad hoc. But I think that here, what is so nice about this type of research is that everything is motivated by, I think in research in general, you're so much more directed, and the math that you learn has so much more meaning, that I think that my mathematical understanding is being enriched, if anything, in that way. Maybe if I didn't even learn new math, which I am, but even if I didn't, I think that it would be deepened in that way, because now I know why. I have some context to put it in. But I also am learning new math.
Thank you, Christy.
Thank you, guys.
And our next speaker is Josh Goldenstein from SDAL. He's a postdoc there. So now let's rock and roll.
Can you hear me? All right. So I'm a postdoc at the Social and Decision Analytics Laboratory. I'll be talking to you about my research at the lab on the applications of emergency management analytics in Arlington County. So this is work that's done in collaboration with the Arlington County Fire Department. And I'm going to talk about two projects that we're doing in collaboration with them. And in conversations with them, with the fire chief, Mike Gowan, he stressed this idea of situational awareness, which I've defined here, the ability to identify, process, and comprehend the critical information about an incident, knowing what's going on around you. So to be situationally aware is to observe and react to your environment. And so we want to ask the questions of what factors adversely affect the environment? And can we improve our awareness? Can we reduce the burden on the environment through more efficient allocation of county resources? So both of the projects I'll be talking about address pieces of this question. And the way that we're helping them to address these questions is by helping them to develop the capabilities to make use of all the administrative data that they're collecting, and also to help them use additional publicly available data to help address these questions. So our collaboration with them is ongoing, and it's iterative. And I think that's a really neat thing about our collaboration is that we get feedback from them. We give them some results, which helps them to focus the questions that they have. And in the end, we end up providing them something that is really useful to them, that they can really implement. So the first project I'll be talking about is modeling response time to structure fires. And by the way, both of the projects I'll be talking about, work was done as part of SDAL's Data Science for the Public Good program. And this is a really cool program. Every summer we get undergrads that come from Virginia Tech up to Arlington. And they're there for the summer. We break them up into student teams. And they're working on a huge variety of projects in these teams. And each project has a sponsor from the local community. So in addition to the Arlington County Fire and Police Departments, we've had sponsors from New Kent, from Fairfax, from Alexandria, among others. And so this is a very focused, great learning experience for them where they get to work on a huge variety of projects. So we're looking at emergency response times. And if we can reduce these response times, we improve the general safety of the community. We can make allocation of emergency resources more efficient. And so our goal for this project is to identify the factors that affect the response times to structure fires in Arlington County. And so in this map, each dot represents the location of a single incident to which a apparatus was dispatched. The color of the dot is the fire station of origin to that incident. So we can see some fire stations are handling incidents outside Arlington County. Additionally, there will be times when all of the units for a given station are out on call and not available. So a unit from another station will have to travel a farther distance to get to that location. And that's a situation where the response time becomes longer. Maybe we see an outlier in the data because the services are basically full to capacity. So the data that we have is administrative data from Arlington County Fire and Emergency data collected over 6 years. It's a huge data set, and it was not given to us in one nice format. So there was a lot of work that went into compiling this information. We were given separate databases that were collected basically from the CAD, the Computer-Aided Dispatch Call Center. There were apparatus deployment files. There's files that give us information on the response time and the incidents themselves. And all of these were collected independently by the medical and fire personnel on the scene, and these systems were not, in all cases, designed to be linked together. So a big part of the question that they had for us was, can we link all of this administrative data that they have to give a full picture of an emergency incident? And so a great part of the effort of this project was compiling this and giving it to them in a form that they would find very useful. In addition to that, we can find geocoded addresses of the incidents. This was necessary when doing some of this linking. We can also attach information about the local communities. And we get that from the Census Bureau publishes the American Community Survey data, which has household level information about demographic and socioeconomic data. So what is a response time? How do we actually define what the response time is? We can break it out into 3 components. So first we have the alarm handling time. That's the time until from when the call or alarm comes in at the CAD call center to when a unit is dispatched. Then we have the turnout time, which is the time from when they are dispatched to when they actually physically leave the station. And finally, there's the travel time, the time the unit spends en route to the scene. And so the guidelines from the National Fire Protection Agency are that turnout time should be no more than 80 seconds, and that the first responding vehicle should arrive in fewer than 4 minutes. Our initial modeling efforts focused on just looking at response time as a whole across all incident types. In conversations with them, we iterated on this and we focused separately on these pieces. So in particular, we focused on the turnout time and the travel time for first responding units. And we focused just on structure fires. So looking at these pieces separately, we fit models both to the turnout time and to the travel times. For turnout time, we fit a linear model. And so we have effects for the fire station, the apparatus type that's responding to the incident, and we have smooth spline basis that lets us get the smooth estimates of temporal effects. And we do a power law transformation of this response. Travel times are right skewed. For the travel time, we have an additional spatial effect. So we are fitting a Gaussian process, and we're doing that to predict the spatial surface of the time to first arrival across Arlington County. And we account for spatial dependence using a Matern covariance. So this makes the assumption that 2 points that are close together have a spatial dependence in their time to arrival. And inference for this model is carried out using Markov chain Monte Carlo. We have the same effects that we did before. Now we have an additional effect for distance, as well as the spatial random effect. So the first set of results I'm going to show for this project are from our initial modeling effort. So this is when we were fitting response time as a whole. And already we can see a clear signal of the effect due to the station. So this is an effect plot of the station on the response time controlling for these other factors, like the call type, and it's fixed at a response to an incident that's 1 mile away. And so we can clearly see that there are some stations that take much longer to respond to this average incident than others. When we focus in on that, so next I'll show our linear model that's fit to just the turnout time to fires. And we can really see a clear signal by the station and by the apparatus type. It is really interesting to note that in Arlington County, stations 106 and 107 are 2 stories. So they actually have a fire pole that you have to slide down before you can get out the door. And so we can pick up a difference in their turnout time here, which is significant, and I find that pretty interesting. We can also see a temporal signal by the turnout time. So this is by the hour of day, and 0 would be at midnight. So there's a higher turnout time around midnight and in the early morning. And whether that's due to having fewer personnel or people are just sleeping or not as alert then, I thought that was an interesting effect. And for the travel time, so this is the median fitted spatial surface from this Gaussian process. And so obviously distance has a strong effect in travel time, but after controlling for that, there's also a very interesting spatial signal that we can see. And this can help us to identify areas of the county that are potentially more vulnerable. And so giving this feedback to Arlington County, to the fire department, it really helps them kind of focus on areas and try and understand what are the root causes that result in these differences that we observe in the response time. So I'm going to move on to our second project with the fire department, which is predicting residential smoke detector need. So here we look at a different administrative data set that's collected by the fire department. And this is a program that they run called Operation Fire Safe. And basically during the summer, on the weekends, they go out and canvass some addresses in local neighborhoods, go door to door, and they check for the presence of smoke alarms and make sure that your batteries are installed and you have as many smoke alarms as you should. And so they've recorded information on over 5,000 of these visits over the last 2 years. 1,700 resulted in actual contact with the occupants. And of those, 31% required some type of intervention, whether that's installing a smoke alarm or replacing batteries. And so our goal, they came to us and wanted to know if we can identify the characteristics of homes that need these interventions to increase the efficiency of their program to help them target their program. And we're doing this using their information over the last 2 years, in addition to some publicly available data sets. So the data that we have here is the administrative data that they've collected. We also have administrative data at the household level, and this is publicly available real estate tax assessment data. So this gives us information for the over 60,000 single-family homes in Arlington. It gives us the tenure, the age and value of the home, the size of the home, and the number of bedrooms, et cetera. We also geocode to get the addresses, to get the longitude and latitude for the addresses of these single-family homes. And we have information at the community level as well. And again, from the American Community Survey, we can get demographic and socioeconomic data by census block group. And examples of those that we used are information about sex, race, income, if you're in the poverty level or not, if you are receiving SNAP benefits, food stamps, your family type, if you're living alone, and your education level. So all of these are potentially predictors that we want to test if they correlate with smoke detector need. So we built a predictive model to predict regions of the county that are in need of smoke alarms. And so the model that we use is a Bayesian logistic regression model, and it has conditionally autoregressive spatial effects. So here our response is binary. It's one if their household is in need of a smoke alarm intervention. And we have a logistic expression for the probability of needing a smoke alarm intervention, PI. And the spatial component is given by this CAR specification, where W is a weighted neighborhood matrix that's capturing spatial dependence. And so if 2 households are in the same block group, then the weight is 1. If they're in block groups that are adjacent to each other, the weight is 1/2. And otherwise, the spatial weight is 0. And we can evaluate the predictive performance of this model using precision and recall. And we do this as we vary threshold. Essentially, we're creating the ROC curve here. We find the value of the threshold that maximizes the precision and recall. We take holdout samples. Our test set is of size 10, 15, and 20%, and we do that repeatedly for each of them. And so for each holdout sample, we fit the model to the remaining data and assign probabilities to the holdout sample and test the precision recall metrics. And we find that for a 20% holdout sample, we're getting 70% of both precision and recall. Additionally, so we had fit this originally for 2015 data. When we got the 2016 data in, we added that to the model. We first used the 2015 data to predict 2016, and then we combined both and used it to predict the households for 2017 data. And so we can get the predicted probability at the level of the individual household, and we can average up to get the probabilities by neighborhood, by census block group. And so our findings for this, we can find which of these predictors are significant for predicting smoke alarm need. And so at the household level, we find that the older a home is and the lower the home value is, the more likely it is to need a smoke alarm intervention. At the neighborhood level, census block groups that had lower median incomes, fewer family households, and a higher percent of people living alone were more likely to need a smoke alarm intervention. And so we use these predicted probabilities by block group, and we get a list of vulnerable neighborhoods that we then gave to the Arlington County Fire Department, and they have used this to target their visits for the summer of 2017. And we are waiting to hear back from them about the results of their canvassing and if it did indeed improve the efficiency of the program as we predicted it would. And we can then update our model with them and continue to help them target their interventions. And so to wrap things up, we're excited to continue working with them. We're also going to expand our work in this area because very soon we're going to be working with the Virginia Department of Health, the Office of Emergency Medical Systems. So we'll have emergency medical response data for the entire state of Virginia, as well as trauma care data. So I'm ready to be grilled. Thank you.
Oh, who wants to grill first?
It's cool, thanks. So I have a couple questions. I'll just ask them both. I have to remember them, though. Oh, yeah. So across applications like this, I mean, have you developed novel or new methods associated with this particular kind of data integration? Are there new statistical methods, analytic methods that are, new? And that's the first question. The second question is related to it, and it has to do with, so as you get more and more problems and stuff, I mean, have you thought about, especially if there are new methods, but even if they're not new, how do you deliver this stuff to people that need it, other than you hand them a list or something? I mean, how can they ask their own questions? How could this be transformed into where are houses that are, need to look at water quality, or any number of things that you might imagine from completely different data sets, different problems, but they need a map like that, and they need the same kind of information, and it has somehow the same. So anyway, those two questions are sort of related, right?
Okay, let me answer the second question first, since I remember it. So I think that the way that we make this useful is we don't, first I'll say we don't take their data and then fit a model to it and call it a day, right? This is a very iterative process. We're working with them, and part of that is we're developing data products and techniques that are going to be useful to them over the long run. So being able to join their administrative data and let them know how they did that so that they can get data analytics people to do that for future data that they collect, I think is the goal of helping them to do. So we want the communities to do their own learning, right? We want them to be self-sufficient with their data. In terms of new statistical methodologies, for your first question, there isn't any new statistical methodology here. I think that the main contribution is really the joining across both all their disparate administrative data sets as well as the found other publicly available data, and using that with some standard statistical techniques to give them information that they would not have otherwise been able to get with all of these separate data sets. Sally?
Josh, why don't you mention, in these two examples, there's not necessarily new statistical techniques, but
Absolutely. So there's certainly, yeah, I'm doing some new statistical techniques for other projects, such as with the Army Research Institute, where we are, again, linking all of their administrative data and fitting a novel survival model that is able to capture the millions upon millions of records observed over many years. So it's not all, there is exciting statistics going on at the lab for sure.
So, but Josh, I was thinking specifically about the different geographic, moving across geography. You're showing one geography here.
Oh, yeah, okay. So yeah, so another application that's more directly related to this is a lot of times when we're collecting this information, it's aggregated at different spatial levels. And so a useful tool involving generating synthetic populations that I'm working on now allows us to move between different spatial aggregations in a statistically rigorous way that is more than what is often currently done, which is kind of using this proportional allocation method. So there are definitely some exciting statistical methods and tools that are used that go into doing this sort of analysis.
Okay, we have our second griller.
So in your first presentation, you had that map of the response time to the fire, and that was controlled for distance. So the fact that distance may take longer to get to.
Mm-hmm.
But even after controlling for that, it looked like regions that were more distant still had a higher response time going into that even controlling for distance. I'm wondering why you think that phenomenon happened.
Yes, so even the residual spatial effect shows vulnerable areas that are away from fire stations, right? And there's possible explanations for that. Are that so the fire stations are put in population centers, right? So it's easier to get from one place to the other in those areas. The map that I showed, by the way, is actually the predicted spatial service, so it is not the random effect. But the random effect does show vulnerable areas in the Northeast as well. So that is a possible explanation.
Any other questions?
Yeah.
In your first scenario, how did you define distance? Was it crow flies?
So distance, yes. So distance, the way we defined it is as the crow flies. Obviously, a good improvement to that model would be to use more detailed transportation information, right? We get NDSSL to help us out with their road networks.
Any other questions?
So in the first one, like, is there, you really not asked the question about right-sizing these different units, like, if more fire incidents are happening in some particular neighborhood, should we pick up that particular fire station than the other one? So like redistribution of resources, something like what Uber drivers do decentralized, like there are more demands there, there's more Uber cars there.
Yes, so what are the possible ways that they can address the resource allocation, right? So,
And just a follow-up,
Mhm.
so again, like, do they track data on what was the cause of fire, and would that lead to like preventative measures rather than like response kind of measures?
so I think that the smoke detectors is one very obvious way that we can focus on prevention, right? So I think that's definitely the case. I think that, what was your first question?
First question was resource allocation.
Yes, so resource allocation, I think that obviously some of the things they can do are to move apparatuses around, or if they're adding new apparatuses, they're very limited in what they can do by the county, obviously. So I think that if they're thinking about adding a new fire station, that's going to cause all sorts of problems and may not actually come to fruition. But I think that if they can sell the idea using a data-based evidential approach, maybe they can get some more traction for resource allocation.
Yeah, so just
I think, Stephanie, do you have something to add?
Well, I'll ask you a question that Kevin just asked me, but then he had to leave. Is
Yeah, so I expected during the time of day for the travel time to pick up a strong spatial signal by rush hour. For whatever reason, we don't see that at all. The time of day for the travel time is essentially flat. So that was very puzzling. I think that deserves some more investigation. Mm-hmm.
Okay, I think it's about time to have another people to be grilled. So our third speaker is Yi Hui Ren, and he's a postdoc from NDSSL. And your turn.
Okay, hello everybody. Oh wow, it's really loud. My name is Ray, if it's easier for you guys. So I'm from NDSSL, and I'm going to talk about network reliability. So if you heard of network reliability somewhere, it's probably different. Wow. So NDSSL, so the fonts changed. And my network is gone. That's interesting. Okay, it's very challenging now. NDS stands for Network Dynamics and the Simulations. So what network is, there are supposed to be edges between the edges between these points. So networks is a mathematical abstraction of many real-world systems. For example, if you have a Facebook account and your friends and you have these links. So it's more like a graph. And when you want to describe your network there are only nodes and edges, but you can also assign or describe every node with some states. For example, if someone is sick or someone is not sick, for example. So the whole network you can denote it as a vector, which is we call that configuration. Okay, so the network dynamics is describing how this network evolves, how these states change. For example, if this middle guy is not well, and in the next time step, this guy got infected and this guy didn't. Or you can think something like maybe your political opinions changed over time, right? So therefore, we have many different network models, and the network is the change of this network configuration is like a Markovian stochastic way. So it's actually a probability. So the next time step, which configuration the network is going to be, depends on the current configuration, and there are some parameters. Maybe some of you feel it's very boring now, but believe me, it's only the first slide. So go. Afterward, it should be much easier. So it has a lot of math. You need to compute, and there's a quiz. So the faster you can do, the more time we can go to the question sessions. Okay. Okay. So the simulation part is the most interesting part in the SSL. Basically, this is very, very difficult to compute because the number of configurations grows exponentially with the number of nodes in the system. So this is really difficult to compute analytically. So in the SSL, we usually use simulations to estimate this probability distribution. And in this talk, I'm going to introduce a particular simulation technique called network reliability. And in the end, you will know why it's called network reliability. To solve a particular problem called sentinel placement problem. But it can solve so many problems. That's why I think this is very exciting, and I want to share with you. And that's in hope you may use it in your own research problem. so the problem is this. So Samartha is a cool guy in our lab, and he has a lot of interesting questions. So this is one of those. So if there's ongoing epidemic, how to choose people to survey, and in order to figure out the transmissibility X or any other parameters of describing this dynamics. So, all my edges are gone. Now I'm worried. So they put this into a math term. Okay, so assuming this is there's a network G with the vertex set, E is the edge set, and we know the source is S. So we know the network topology and also the source node. The question, we can divide this question into two. One is given the states that you observe. So you know this guy is the source, and after some time you know this guy is healthy, this guy is not. How do you compute the transmissibility X? So what is the transmissibility X, allow me to explain, is if this guy is sick, what's the probability along this edge this guy fall ill? Okay, so that's transmissibility X. So the second question is, at any time moment, where do you to put sentinels, or which people do you to pick to survey to help you to answer this question better? Okay, I know it's really a little bit vague, but after you do some math problems, you will understand better. Oh, sorry, I lied. There's another slide. Okay, it's actually very good. Network reliability. The first problem is actually an inference problem, right? So given the observation and the network, you want to infer what's the distribution of X. So if you can draw the curve of X, then you can say, okay, the max argument max, which is the most probable X, is your answer. The tricky part of the stochastic system is even if you fix the X, if you go forward, the configuration can be very different. But if you observe which means if you observe the configuration, if you trace back and figure out what X is, it could be any X. Any X will give you the same configuration. So that's the tricky part of this problem. We think that this can be solved. I mean, we can release the network reliability, or we can form this problem as a network reliability problem. So what this means, this Rx, Rg means is the probability that observed configurations occur in the graph, the probability of that occurs. So where X is the transmissibility, R is we call it criterion, or with the configuration is consistent with observation, and G is the network. So once you observe something and the graph is fixed, these two R and G is fixed. So you can write this probability as a function of X. For in case you want to know more, this is how we do simulation. Because to compute this is the Sharp-P hard problem. So basically, we will talk about more about this during the question part. But we use Monte Carlo simulation to simulate PK. And for those people using Illustrator pen tool and wondering why this thing goes this way, so this is the math behind it. It's called Bernstein basis. This is called Bezier curve. Okay. Okay. So now is the math part. I'm glad my edge is coming back. This is very simple, right? So if this guy is sick and this guy got infected, and given what this is the probability that this configuration happens, right? Because this is the probability X, and it's the simplest case I can imagine. Okay, now is your turn. So given this node is the source and both people got infected, what's the probability of this happening? Qijun? Exactly. Very simple, right? So I can see we can go really fast. How about this one? Anyone? So if this, okay, this green one means unknown, right? You don't know the states. So if you go from upper one is x squared, right? If you go to lower one is x squared. And while you double count it, if you have to reduce the double count, so this is called inclusion-exclusion principles, right? So it's actually 2x squared minus x4. Everyone get it? Okay. So easy. But as you can see, it can become very, very complicated once the graph have a lot of loops, a lot of things. So it's really complicated. But we can do this for small network. How about this one? So we change this infected people here, right? So anyone want to give a try? So it's x cubed, and the minus x4, right? So which means when we're observing the epidemics, we are not only not only the number of people infected is important, but also where they are infected is also very important. It contains information. So, but it's not really helpful to answer the question we originally proposed, which is to answer what x is. Because if you see the argument x, argument max x is always 1 for this, because it's monotonic increasing. So it's not very helpful. What if we know where the healthy people are? So the green is healthy, so it's 1 minus x, right? So if it's 2 people are healthy, it's 1 minus x squared. And here's the interesting part. What if one is got infected and another one is healthy? Right? It's x times 1 minus x, right? Now you have something to answer, right? So now we know the arg max is 1/2. Okay? So let's make it slightly a little bit more complicated. So let's add one more node. And so in this one, this guy is infected, this one doesn't, and this one is not, and this one is infected. It took me a while on the paper to put it down, so I won't ask the. So this is x and 1 minus x and 1 minus x plus x. So you see the maximum changed. So which means if you observe this, this is the answer you gave. And if you observe this configuration, this is the answer you gave. Okay? So basically, the first question is solved. So once you observe something, you can use the network reliability, which is the equation I didn't explain in details. You can use that to give you what is the x hat is. X hat is the argument max. Any questions? No? Okay. Good. Yes? The middle one? Right. So in reality, we use the computer to do that. But you can do this with the pen. So this one is definitely infected, right? So that's why they have X here. And these two events are independent. So X is always there. And this one is interesting because this one is not infected. There are two cases. One is this one is not infected. Another one is this one is infected, but this edge failed. So you put them together, that's the answer you have. Okay? Good question. So the second question is very, very interesting, is where to place the sentinels to give you the best answer. So because if you only have to, this is observation, this is x hat, but for any x, it can goes to this configuration with exactly that probability. So now, if we have 3 candidates or 3 sentinels for this graph, and we have 8 possible outcomes, right? And I just decoded them binarily. So the 0, 0, 0 means everybody is healthy, 1, 1 means everyone is sick, and everything in between is there. And you can do exactly the same thing as we did before, and each of them give you a curve. And the yellow point is the answer you will give if you observe this configuration. So what I'm saying is, if you treat this x hat as a random variable, and you can see what's the distribution of this, and you need to put you see these two configurations give you exactly the same x hat. So you need to kind of put them together, and that's why the sum is. This figure is just a different way to draw it. Oh, okay, that's surprising. So these are points. So this is the plotting the distribution of x hat. The question is how much information this distribution provides, right? So if you choose a different set of sentinels, you will get a different set of this. You will get a different distribution of x hat. The direct answer is entropy. So entropy is the average of information. So that's basically, once you ask this question, if you know entropy, it takes very few seconds to answer. Okay, sorry. So which you put the numbers here, this gave you 2.188. What that means, it means if you have 3 coins and you flip, and they are independent with 1/2 probability, this number should be 3, which means 3 is the upper bound. So because and we will know why this is the case. So here is the real question: is what happens if budget is equal to 2? So you have to choose. Thank you. You have to choose 2 out of 3 of this network to do the best detection. Okay. Here is the all possible 3 ways. Apparently, this one is better, so it's 1.8. And let's see why this is. Why this is? Sorry, there's a lot of information. I know. Why this is? It gives you 4 curves first, right? And it gives you 4 different values. So your distribution have actually 4 different values. That's why it contains more information. And why this one and this one is not that good, let's say this one, because it gives you 4 curves, but 2 of them are overlapping because it's symmetric. And for this one, if one is not infected, you don't need to ask 2 because it's dependent. 2 states of 2 depend on state of 1, so you get one which have one of the curve is 0. And among these 2, you see this is slightly worse because this is a uniform distribution, which is the maximum entropy, and this one have 3 points, but it's not uniformly distributed. So what I want you to take away from this is when you're selecting sentinels, it's better to be independent, and it's better to be asymmetric, so you have more values. So this one is symmetric, this one is dependent. Okay? Now is the quiz time. Oh my God. I don't really know how to do this, but this is if you know K is the source and the edge connects from A to B and B to C. Okay, so it's like a tree. There's no loop, and we pick 3 nodes to maximize the entropy. Okay, and I can give you a little hint that the network you see all the time is this one. Okay? Remember the rule, independent, asymmetric.
No, I didn't understand the second word, the shaded thing means.
You see this network? Yeah, because we don't have the edge anymore. So that's it's not fair because we don't have the edge. So the answer is this one because it's not in any of these 2 branches, but we don't have the edges. So let's go to the application. There are one application is really direct. So we have School 86 is the code name for high school, and we have Friendship Network. So if we say the middle is the guy infected, so the green ones are the sentinels we want to place. So, and we can do the similar things on another network, which is World Airline Network, and Hong Kong is infected. So this, why my edges are all gone? What? That's really, really, really strange. Because this is baked into the PDF, so I don't know why it can be removed. Okay, I'm so sorry, but you can imagine they're all popular, not popular, but central airports, and they have a lot of edges, and that's really weird. Okay. Now you may wonder why the name network reliability have anything to do with what I'm saying, right? So network reliability was first proposed in 1956. So the topic they were studying is how do you build a reliable circuit using unreliable or less reliable relays? Because at that moment, everyone using phone, and there are not only one phone lines connect to 2 points, there are a lot of redundancy. So Shannon and Moore have these very nice articles basically talking about the failures of the relay and what's the probability connecting from S to T. And Stephen realized, okay, this is really powerful mathematical framework rather than just a relay, but we can also have a attack rate because a failure of the relay is the inverse of getting disease transmitted. And from there we have no chance passing. We have a lot of interesting things, like we can connect to the Ising model in physics. We have characterize the ERGMs, and the sentinel placement is this talk. And I hope you have you can have some interesting questions and see if we can apply these methods to more problems and their teammates. And yeah, thank you.
Nice talk, and let's make Chris the first growler again.
Medium well, please.
So do you have do you have any kind of theorems that say something like for graph properties, something, size of graph, edge density, whatever the properties might be, yeah? That there's a bound on the number of sentinels needed to have a certain level of confidence that, you know, are there any bound style theorems?
I guess what
I mean, as a theorem, I mean, is there something that we can read that like does that?
No. Let me think. Let me go to this. If I understand your question, so here's the thing. In network science, people tend to characterize the graph by its topology. For example, the density, the clustering coefficients, and et cetera, et cetera. And this way is actually combining the topology with the dynamics and configuration you are interested in. So it's not only a measure on the topology, but combined. So it's a more direct way. And to answer the second kind of second part of the question is if there's a theorem, because apparently this is really, really difficult to compute. It's a Sharp-P problem. It's actually, we have this error bound. When you do the Monte Carlo, basically the question is how many Monte Carlo simulations do you need for different size of the graph? So if the graph is dense, there are a lot of edges. And this n is actually the number of edges. So essentially, every time you draw a sample graph, it's a Bernoulli trial. So it's either consistent with configuration or not. So therefore, the error bound is literally the Bernoulli trial's error bound.
So my question
Oh, Sentinel's foot.
Bound on the number of sentinel measurements that you have to be able to take in the first place to know that you're measuring anything, right? There's some number. You can't measure everything.
Right, right. Oh, I thought I was totally misunderstood.
Is there some bound on that number?
No. The answer is, even though you sentinel everybody, you don't know your X for sure. But the more sentinels you place, this curve becomes narrower. So the more sentinels you place, this curve becomes more narrower. In that sense, you are more confident that the X hat you have.
Sure, but that's qualitative. What's quantitative?
I don't know.
You put 5 more in, you get what kind of more concentration of your X curve, right? So you want to have the sweet spot. For what graph topology do I have to populate 50% of the nodes or whatever a recursive algorithm is going to assign sentinels in some kind of fashion? So what do I get? What's the quantitative relation?
I'm not aware of. But it's a very good question. I should think about it. But computationally, this is very expensive.
No, I know, but
yeah.
Sometimes you have very powerful approximation algorithms, right? Even though you have sharp-p-hard problems, right?
It's more than sharp-p-hard, yeah. I don't want to. I guess this is time I go to the.
What's Sharpie? Sharpie is how hard the problem is of finding the number of sentinel
No.
mutations? No.
Sharpie is
the question really is that. I mean, it's a straightforward question. It's saying, if
right.
you're going to put sentinels, how many sentinels? That's all I was asking really, is like how many do you have to put there to get to a certain, there's a relationship between the number of sentinels and some error epsilon. Right? So even if the error is not 0, when everything is a sentinel, it's something. Right? And so the question is, like, how does it converge as the number, I don't know how to state the question because I don't know what it would look like, but I just wanted to know if you knew what the bound on the number of sentinels is, like, sampling style theorem.
Right.
Best bang for the buck.
Right. Let me think about it. I guess what I'm saying is forget about all the computational complexity of things, assuming given these sentinels, this is all the 2 to the n curves we have. If they're big sentinels, we have 2 to the b curves. And each of them are different. Each of them give you x hat. And you group them together, have a distribution, and it tells you it tells you an answer, right? And the x you are estimating is only be one of those. And the true x, let's say you run a simulation behind this, could be none of those. So there's really difficult to define the error in a traditional way. So, right. So to
Any other questions? Okay, I think it's about time, so let's welcome our last speaker of the day. So our last speaker is Meghna Verma. She's a graduate student from NIMO, and here she is.
Hello. Okay. Yeah. Good afternoon, everyone. I'm Meghna Verma. I'm a graduate student at the Nutritional Immunology and Molecular Medicine Lab. I'm hereby going to refer that as NIML. And today I'm going to discuss the vision that NIML has for precision medicine and health. I want to start by thanking my team for the useful feedback that they have given me along the way. Also, I want to highlight that we are a group of people composed of immunologists, mathematicians, computer scientists, and engineers. I'm an engineer myself, so we really work as an integrated unit towards solving the challenges in human infectious diseases. Okay, so NIML uses a unique iterative cycle to study the biological interaction system. So what we do is we combine advanced computational methods along with experimental technologies to study human infectious and immune-mediated diseases. So we start off with the hypothesis formation, and then we progress towards performing in silico experiments. We then go on and use the knowledge that we have derived from the experimental methods to create computational models. And the technologies that we use include the high-performance computing capabilities of BI. Then we go on and analyze the model with standard sensitivity analysis methods with the hope to get new emergent properties or new mechanistic insights with the model that we have developed. We then go back in the lab, and the findings that we have obtained from those models are then validated again in vivo. And the cycle continues by checking like whatever new therapeutic targets or new findings that we have found out. We try and check if the things have been implemented on the clinical trial level. Yeah. So I would like to focus your attention on the graph on the right side. So at NIML, we model the biological system at different scales. It involves an intracellular scale, intercellular scale, cellular scale, molecular scale, and also at the tissue level. In the future, we hope to take this to the population scale level, wherein we plan to conduct like in silico clinical trials with the findings that we have obtained from the experimental data. So really the model integrates the findings at different scales in the biological system. This is just a timeline of all the models that have been developed since 2010. The models include a wide range from modeling inflammatory and immune-mediated disease in the gut, such as inflammatory bowel disease or Crohn's disease. We have also modeled infectious diseases, including Clostridium difficile, Helicobacter pylori infection, the one that I'm going to talk about in more detail today. And we have also recently modeled the human immunodeficiency virus and human papillomavirus co-infection model. Okay, so today I would like to talk about this important problem, antibiotic resistance, which is not only a security but an economic threat. The graph on the left shows that a recent report on antibiotic resistance highlighted that if the problem is not dealt or if we do not find new antibiotics, overall antibiotics resistance can cause about 10 million deaths by 2050. And that you can see is clearly more than the people who die currently of cancer. That accounts only up to 8.2 million, and even diabetes, which is like just 1.5 million. So the difference is pretty large with the amount of death that can be caused just because of antibiotic resistance. As I mentioned before, it also accounts to the economic threat. And the report highlighted that up to $100 trillion would be the part of economic loss that would be caused just alone because of antibiotic resistance. So knowing this problem, today I'm going to focus my talk on one of the pathogens that the lab is studying, which is Helicobacter pylori. Recently, in 2014, the World Health Organization classified Helicobacter pylori as a high priority pathogen, meaning it was identified as a pathogen for which, due to antibiotic resistance, we urgently need new treatments that need to be developed in the future. And just a little bit more information on Helicobacter pylori. It colonizes and infects the stomach. And the scale on the right shows the pH scale for different acidic substances. And you can see that stomach has a very high acidic environment. So this bacteria has found ways or has adjusted itself in such a way that it tends to thrive in such a high acidic environment. It is also known to be a major cause of gastric cancer and ulcer. However, there's recent evidence of protective role by Helicobacter pylori in diseases such as asthma, inflammatory bowel disease, and obesity-induced illnesses, wherein there have been studies that talk about when a person is infected with Helicobacter pylori, they have been shown to be protected against the diseases that I just mentioned. Furthermore, our lab recently published a study that highlighted the regulatory role of H. pylori in the stomach. And what the study found out was that there was a specific subset of immune cells, meaning the defense system of your body. There's a specific set of cells, hereby I'm going to refer that as macrophages, so not getting into too technical details of that, but it was shown that these cell population kind of cooperate with the bacteria in order to make sure that the bacteria is flourishing in the stomach without showing any inflammatory mechanisms. So overall, as I mentioned before, H. pylori is known to be a major cause of gastric cancer and ulcer, but due to the recent evidence of the regulatory role, the dual role of H. pylori comes up as both being a commensal and a pathogenic organism. There are current therapies that target 3 levels. The first therapy level targets the host, wherein there have been immunoregulatory products, including the vaccines, that deals with the infection at the host level. There are therapies that target the stomach, that is the site of infection. And the third level of therapy includes targeting Helicobacter pylori itself, so just like trying to eradicate the infection at the bacterial level. In our lab, we focus at host-based therapeutics with overall a precision medicine-based approach. The reason being, we believe that if we are able to better understand the immunological mechanisms at the host level, we will then be able to better pinpoint like how these mechanisms, or like what is the genetic variability, or like why is the bacteria showing a different response in certain individuals as compared to other ones. And a part of doing that is, as I mentioned before, we do believe that use of computational models can definitely facilitate this complex dynamic interaction between the host and the bacteria. And we use a complex systems biology approach, wherein we have integrated ordinary differential equation-based model combined with the agent-based model framework. And some of the main features include that because of the stochasticity of the simulation in the agent-based model, we are making sure that the biological variability has been taken into account. And we integrate this ODE-ABM-based framework with the previously published model that we have in the lab for certain immune cells. This is just an example simulation wherein we have modeled the different compartments in the stomach. So there are 4 compartments that we are looking at. So the lumen, epithelium layer, lamina propria, and gastric lymph node. These are the 4 compartments in the stomach that we are zooming in. And the small circles and so the small red circles on the bottom in the lumen are the Helicobacter pylori that we are looking at, and the epithelium layer, the squares that are turning from blue to red, is basically the layer of epithelium that is getting compromised because HP is destructing that layer and trying to get into the different compartments. Furthermore, the other circles that we are looking at are nothing but the immune cells that we are modeling using ordinary differential equation-based model. Okay. So having this framework in place, the main questions that we are trying to tackle include looking deeper into what are the mechanisms of this subset of population that we recently identified, like how are they exactly playing a role with HP in facilitating its colonization in the stomach. Furthermore, we are looking at what different chemicals these cells are producing and how is that contributing towards the infection. And with standard sensitivity analysis-based methods, we are hoping to find out new unforeseen emergent properties of the system so that we can focus at things that we have not looked at experimentally. So we are hoping to find like new insights with answering those questions. So it's just a highlight of the 3 questions that I just mentioned, just a few results from that. Obviously, I cannot go into all the details. So this is just an example simulation where we had 2 groups. The second one, the PPR gamma knockout, it's basically a gene that was shown to have a protective role in the infection. So we in silico simulated a gene knockout model and compared that with the wild type simulation. And we observed that there was a differential response in these 2 groups. In terms of looking at the chemicals that were produced by these cells, we were also able to achieve that using our computational model. So we were able to pinpoint like what cells are producing these chemicals and how much of these are being produced in different conditions. One of the interesting things that we found out was there was a parameter that we were not previously looking at on the experimental side of things, but currently the model seems to direct us towards a new direction to investigate. So we are planning to look into that into more details. This is a picture of the mechanisms of immunoregulation that we studied in a pig model of H. pylori. So this is just to give you an idea that the experimental models that we study at NIML include mouse models and pig models as well. Pigs have closer immune system similarity to the human system. So the red spots right here are the H. pylori in the gastric mucosa, and the one on the bottom are the epithelial cell layer. So that's like a layer in the stomach that HP disrupts in order to enter inside. So overall, the methods that we use here have or can be applied to develop or test new therapies, or at least move towards a direction where we can create new host-based approaches as compared to targeting the bacteria. Since currently, whoever is infected with H. pylori, the physicians just would prescribe you an antibiotic treatment. However, with these computational approaches, we hope to create an in silico patient avatar or few models wherein the treatment can just be tested even before like prescribing antibiotic treatment. So that would be more host focused as compared to just using an antibiotic, which is generally prescribed. And overall, the combined experimental and computational approaches can help us lead towards new predictive insights that we would not have investigated only by experimental methods. And this combined approach is very well applicable to other infectious diseases as well. So overall, our message is we are moving towards an era where we hope to achieve a personalized, predictive, and a preventive approach towards treating human infectious diseases. With that, I would like to thank my advisors, Dr. Joseph Bazagania Riera and Dr. Raquel Hunter-Silas for their continued guidance. I am in the Translational Biology, Medicine, and Health program, and of course, the Biocomplexity Institute to give me this opportunity to come and share my work with you guys. Thank you.
Who wants to grab the chance?
Everyone is tired.
Any questions?
So in this particular setup that you particularly are talking about here, not in general, but in this particular one, so you have all these different layers and models and so on. So how do you handle the question from the point of view of, say, somebody who would be interested in trials, right? How do you answer the question about how component-wise verification of these systems lead to system-level validity in the results that you find? How do you approach that question? Do you understand my question?
I think so, yes.
Okay.
You can correct me if not. So with these approaches, translating it to clinical setting, I think that's what you're asking, or...
No, that's what I'm asking at all. Here, I'm saying those would be the kind of people that would... Those would be the kind of people that have this question, but that's not what I'm talking about. It could be anybody, it could be me, it could be somebody else. So you have a very complicated collection of models. They're very different, they have different data sources and so on, right? So in this particular way of sticking things together, in this particular way, you know, there's always the question of validity of a model. I mean, you know, this is like a real pain in the neck question to ask. This is related to that question. It's not exactly that question.
Okay.
It's a specific form of it. How do you approach the question in this system with all this collection of data and models, right? How do you, as you think about building one of these things, how do you approach the question of validating your results? How do you know that these models are producing reasonable outcomes? How do you approach that question? Not whether can you do it. I want to hear about how you do that, what you think about when you build these to ensure their reasonableness and their validity. How do you think about that formally or experimentally, or however you might do it? Do you understand that question?
Yes.
Okay.
Okay. So the way I would approach that would be, if I already know how a person is responding, if he or she is infected with H. pylori, we can probably collect data from that person, and in general, like whatever we know about the model that we have developed in H. pylori, we would then try to fit that data with the current model we have. So that way we will know, like, okay, there's a model in place which takes into account all the immune responses in a certain host, and then we have this data that we have collected from a infected patient. So using that data, probably that can be an approach where we can fit the model with the data that has been collected, and we can look at the responses, like what's exactly the output from the model once we have that data in place. And we can compare that to our previous findings and try and see what are the things that are missing. So that would be like an approach wherein we would go collect data from the patient and then fit that in the current model and then compare if there are certain things that are not consistent with what we previously had.
But wouldn't the goal be to try and predict the therapeutic outcome, not to just fit the therapeutic outcome? I mean, the goal is not to fit data, right? The goal is to use it prognostically or at least diagnostically, right?
Probably not diagnostically, but maybe, yes, predictively. So the reason why I talked about like fitting the data would be having a baseline and then using it, like once we know that the data fits well with one particular patient or a certain group of patients, it can then be used as a predictive method for the other group. Yes, some are useful.
You can always drive it and improve it. The model is not an outcome, it's a process.
No, I know that. I'm not asking you whether it's valid or not. I'm asking how you think about using it. You know, obviously you could make it. I'm just asking how you think about it. In the settings that it's going to be used in. I'm just interested in how you approach the question. Right? I'm not challenging whether or not you can call any model wrong or any of that generality. I mean, in a particular setting, an infection setting with a collection of patients, or maybe this particular patient, or you have some data and a new patient, I just wondered, you know, is there a process, like a protocol, a thought through perspective on you know, how to use it in that way, so that you get to the place that you say, well, I mean, if we did this, it'd have a higher likelihood of having a good outcome as opposed to that. You know, if you know, whatever. Anyway, it's just that's what this is for, is to ask that kind of question. I mean, complicated models are always hard to validate. We all know that. That's not the issue. We all know that. We all sympathize. That's not the issue. I just wanted to know if there was sort of a perspective on how to do it, and in place, or maybe even built into the platform or something. But anyway, that was what the question was.
I think we have time for one or two more grillers. So.
So I have a general question, not just like first I see NIML as one of the labs that it's an individual health, like as a like NDSL looks at like population health, modeling organ systems, and looking at specific diseases. And so, I mean, this is something that I've asked people even inside of that. Like, we have tools which, in the context of population health, infectious diseases, can simulate the progression of the disease without interventions, and then try out different interventions like school closures and vaccination, and try to give a intervention evaluation kind of a platform, like civil is one example. So just an open question of how long do you think it would take for precision medicine and all these approaches to get to there for an individual level model? Like, can you take an individual, do diagnostics, get statistics, and then do disease progression modeling, and then take all the therapeutic methods, like new methods, well-established drugs, and then put them in, and then test out, and say, okay, this is what I think is the best intervention now, and this is how I think this particular metric, like the population of Helicobacter pylori in your stomach will grow like this if I do this now, instead of doing this 15 days from now. So how long do you think, or is it in the horizon, or is it at least?
So we have actually worked on something similar that you're talking about, but in a different disease infection, that's Clostridium difficile, where what we did was we created like in silico patients. So we had a ordinary differential equation based model for C. diff infection, and then we looked into the clinical trial data. That work was done by a previous graduate student, Andrew Lieber. So he collected data from a clinical trial study of all the treatments that were known for C. diff. So those clinical trial data had information about, like, specific information about each individual patient who were given these treatment options. So what he did was he took that treatment information or the effect of those treatments, and then he applied that to the ordinary differential equation based model, which is similar to saying that, okay, you are using treatment A, B, C, or X, Y, Z, whatever. So you're using that in an ODE based model. How is your output different? So that kind of maybe partially answers your question, meaning like what is the effect of the treatments? I think we are there, but in terms of collecting all the data, I think it's a data analytics or a big data collection problem, where obviously the future is, and we hope to move in that direction, where we collect all this data which is particular to a certain individual, and then combine that with the known treatment effects together, which can then be applied to this model. Does that answer your question?
It seems that no one wants to be the last griller, so I will be, and I will make a happy question. So, relax.
Thank you.
So basically, I know you are still a graduate student, right?
Yeah.
So I want to ask you, what is your future plan, and what role does your group and this entire BI plays a role in your future?
What is my future? Like,
Yeah.
what is my plan for the future?
And what do you learn? Like, what kind of experience in BI and your groups helps you to pursue your future dreams? Yeah.
I think it fits pretty well with what I want to do next, which is probably work in again in a research setting, more applied towards human health, maybe a hospital research organization, and the skills that I know I would require would be data analysis and data management and like computational modeling. So coming from a biomedical engineering background where I did not do all this work that I'm doing currently, it definitely helps put things into perspective for the future steps that I plan to take.
Let's thank our speaker and
Well, that was really interesting. It was great. There's a broad range of work going on here. I think I just want to remind, in case there's anybody here that doesn't know this, these are all samples from these various organizations, and within them there's a distribution as well. So as broad as that distribution is, the actual one is bigger than that. So cool. Okay, so there's in a minute, after I finish, don't play it until I tell you guys. Yeah, don't play it yet. No, wait a minute. Oh my goodness. Wait. Man. So the, yeah, so, okay. Lost my concentration here, so I got to get my concentration back. So the Institute does a lot of things, and we have a lot of different kinds of capabilities here. And I think it's fair to say, not just at this Institute, but I think it's kind of obvious, much broader than our Institute, that the the world has built a new interactum. It's built a new medium that we interact in. It's not virtual. It's real. We interact in it, and it's the ICT layer, right? It's just there. And it's not the same world. It's like saying the stock market before margin trading was just the stock market that we have, but now we have margin trading. It's not. It's an entirely new market. It's an entirely new thing. So I use that example all the time for people that talk to me. So if you talk to me, you've heard this example. But I could make other examples. I can't think of one right now. But there's lots. The point is, in research, where we really are moving in a direction where computation isn't something that a different group of people do than researchers in any other discipline. Computation is more and more ubiquitous, even if we don't think of it as really part of what we do because we're really a biologist, but there is this computing stuff that somehow we have to do. But it's not really like that. I've been at a couple of HPC conferences recently, and in all of them, the HPC community specifically is still sort of focused on a particular kind of problem, like calculating the wind fields over the car body, or around rotor blades, or shocks, or even weather, other kinds of things. And I can't tell you how many talks I saw, at least for sure, one of them that was actually an HPC center, where the talk starts with a screen dump of somebody's VI, page, and they're going through code, right? Which is fine, except for one problem, is what's driving programming is an ever stronger force that reduces the number of programmers that have to produce a given number of lines of code. This number is dropping. Number of programmers per lines of code is just really dropping. It's not just dropping because of automatic programming or something, although that's true. It's dropping because of high-level languages, and increasing abstraction is changing, where things that you used to have to code before, you just go point to it. And it's changing things, and it's going to change how this kind of science is done. And the reason I'm telling you that is that, well, okay, that's one factoid. Another factoid is that when we started here as the Bioinformatics Institute some years ago, about 80% or so of the revenue of the Institute was related to work that ultimately was done in really in wet lab biology and programs associated with wet lab biology fairly directly. And really only about 10 or 15% of it was done that was, centered in computational research, and there was a certain amount of cost recovery sort of stuff that made up whatever the difference is in the numbers I just said. But those numbers are flipped. The numbers are flipped. Now, numbers alone aren't what we do in science. We don't just hit dollar targets on science, but we do realize that this means that the direction of science has changed. It's a signal. And we're very much well posed to do that. We're never going to not need to have experimental work in labs. That will never happen. We will always need to do that. How that is achieved, that may change. But it is true that computationally, things are going to have to become a lot more thought through to take advantage of computational basis from the beginning in order to remain at least competitive going forward in many, many, many areas, not just in the life sciences, like everything. We've built this layer into the world, and it's just part of the world. And so it doesn't matter if you're traffic engineering areas as well as expanding into these more social areas and so on. But we are going to have to repurpose in a systematic way large portions of the Institute facilities over time to accommodate modern science. And so one of the things that's happened, as everybody knows by now, is over the last couple of years, we've gone through a process of converting a growth chamber into a high-performance computing facility. In some measure, it has simply, it's going to allow us to move Shadowfax into a new and smaller footprint because it's water-cooled and you don't have to have as much space between things that you have to have with air conditioning. It's more energy efficient because the water reduces your air conditioning loads. So you put a couple megawatts in, you've got to take a couple megawatts out. And the question is, how expensive is that going to be and how do you achieve it? And you do it with water, bottom line. So that allows us to get to a smaller footprint. It also is expensive. And it has to be done in a way that doesn't, it has to be done in a smooth way. So the bottom line of all of this is that we finally are to the place where we can say that over the last 3 years or so, we've about roughly tripled our computational capacity. We have about a 200 teraflop HPC capability right now. If you just view that part, there's other capability here than HPC, but I'm just talking about that. We have, petabytes of disk. We have lots of things. All this is going to grow over the next 5 to 7 years or so. If you take into consideration everything, the footprint, the number of racks we can still add, the amount of power that's available here without alteration, and the shrinking of computational power on given pieces. If you take all that into consideration, we should be able to go up by a factor of 10 from where we are now over about the next 7 years or so. So we'll never be a national high-performance computing center, but in terms of having a work-a-day, workhorse level HPC capability here, we have that. Okay? And we do have the framework for maintaining a very modern and leadership capability as this kind of research evolves. So what I was saying about the computing thing is that with the high-performance computing capability that we have, what's really important that we make it available for is services, because everybody doesn't want to open a VI window and start coding at that level to get these things to work. So the tools that are being made here, we really do need to think of them as leveraging capabilities that make the HPC capabilities available, that provide access of these capabilities to research. And I don't think we can rely exclusively on other people to do that, and so we're going to have lines of work that focus on that, even in labs that are more sort of intellectually oriented toward the life sciences. And this is going to have to increase. But to that end, today we had the ribbon cutting on the current state of our computing center downstairs during lunch. They want me to play you the little movie of me shuffling around with giant scissors and trying to figure out what to say. And so it's all very embarrassing. Sometimes videos are the brutal truth. It's hard to look at, but there you go. And so, so why don't you go ahead and play that now? Okay, they're not. Hello. Thanks for everybody for coming to this. It's this is our 200 teraflop computing center. Our computing capabilities have gone up by a factor of 3 or 4 in the last 3 years. We have room to grow by another factor of 10 over the next 7, 8 years. Hopefully it will last that long. It's a big step forward for the Institute, and I think it provides a real increase in capability for the university as a whole, and we're really happy to be able to call this done.
Congratulations to the Biocomplexity Institute. This indeed is a significant advance in support of our university's research program, really in service to research at the state level. And so this really makes a difference, not only in terms of how it advances research at the Biocomplexity Institute, but it makes a difference for all of us as we emerge various destination areas, particularly those that really engage the data and decisions destination area. So again, congratulations, and I very much appreciate all the hard work that's gone into this accomplishment.
Okay. So there you go. Quite a work of art, right? Okay, so the one thing I want to add before we go is that's our interim provost. Cyril is a solid guy. He's a good guy. The guy to the left is the vice president for computing and stuff here, IT, VP IT. We're in a really good place right now. Okay, we need to grow. Growth here, we don't need to grow really, really big. Okay, we don't need to be a lot bigger. We need to be a little bigger. We can always do to be a lot richer. So. It's always better to be richer. So don't stop working to become, better funded or whatever you have to do. But we're really in a pretty good place. We're in a much different place than we've been before. It's completely due to your hard work and your talent. I think that it's really, really a hopeful view forward. It's not an easy view forward. It's a lot of work, but I think we're here to work, so. If you don't like it here, if you don't like the work, then you need to revisit what you're doing every day. But there's going to be a lot of it, so you'll have a lot to like. So so I guess that's it for today. If you have any questions for me now, or really any time, please come visit me or call me or send me email. I'm happy to try and help whatever issues you might have, and I'm also happy to listen to whatever you're doing because I'm genuinely interested. And so. With that, goodbye. Come to the reception and receive, I guess. Bye.